Assalamualaikum and hi everyone. My name is Nura Ada binti Nozaid and I will be presenting the introduction of Giardia Duodenalis. In this section, I will briefly explain what Giardia Duodenalis is, its historical background, its binomial nomenclature, and how it fits into the five-kingdom classification system. Giardia duodenalis, also commonly referred to as Giardia lamblia or Giardia intestinalis, is an anaerobic, flagellated eukaryotic protozoan of the family Hexametidae. It is one of the most common intestinal parasites affecting humans and mammals globally, causing a diarrheal disease known as giardiasis. This infection is typically transmitted via the fecal-oral fluid, primarily through ingestion of cysts found in contaminated water, food, or through direct contact with infected individuals or animals. The first recorded observation of Giardia was in 1681 by Anthony van Leeuwenhoek, who discovered the organism in response to samples. The genus name Giardia honors Professor Alfred Giard of Paris, while the species name Lamblia commemorates Professor Václav Lamb of Prague, who conducted detailed studies of its morphology. Though it is referred to by different names such as Giardia lamblia, Giardia intestinalis, and Giardia duodenalis, they all refer to the same species. The accepted scientific name is Giardia duodenalis, where Giardia represents the genus and duodenalis reflects its common location in the duodenum of the host. Despite multiple naming conventions in literature, this binomial name is the most widely used in modern microbiological studies. Giardia duodenalis is classified under the kingdom Protista, which includes diverse unicellular eukaryotes that do not fall into the plant, animal, or fungi kingdoms. The complete taxonomic classification is listed below.
Giardia duodenalis, also known as G. lamblia or G. intestinalis, is a single-cell parasite that lives in a small intestine, especially the duodenum part. It has two main forms, which are tropozoid, which is active and helps parasite to attach and grow in the intestine. For the second form is cyst, which is inactive but strong enough to survive outside the body. For the first one, the structure and the features, tropozoid form. The tropozoid is the active motile stage. Its shape as tennis racket shape with a flattened body that can appear sickle-shaped if we view from the side. It measures around 12 to 15 micrometer long and 5 to 9 micrometer wide. The move used 4 pairs of flagella. The key feature is a ventral sucking disc which helps to attach to the intestine wall. It has two nuclei that give a face-like appearance, axosal for support, and two curved parabasal bodies near the sucking disc. Okay, next for the cyst form. The cyst is the infective non-motile stage of parasite. It is oval-shaped, smaller than tropozoid form, and surrounded by tough outer wall that protect it from harsh environment. It typically measures around 11 to 14 micrometers long and 7 to 10 micrometer wide. It's depending on its stage, it could be 2 or 4 nuclei. For 4 nuclei, it in mature form. The cysts allow to survive outside the host, especially in water, food, and it causes infection when it's been ingested. Nutrition and energy metabolism. Now, let's take a closer look at nutritional characteristic and energy metabolism of Giardia duodenalis. This organism is classified as a micro-aerophilic anaerobes, which means it can thrive in environments with low oxygen level and do not require oxygen for its survival. In fact, Giardia lack of mitochondria and the ability to carry out cytochrome-mediated oxidative phosphorylation, which is the primary method of energy production in aerobic organisms. Instead, it relies completely on anaerobic fermentation for generating ATP. Its main energy source is glucose, which is metabolized through fermentation into various end products, such as acetate, ethanol, alanine, and carbon dioxide. The type of metabolic byproducts depends on the oxygen concentration in its environment. When oxygen levels are higher, ethanol and acetate become the predominant products, while under the strict anaerobic condition, alanine production increases. This metabolic flexibility allows Giardia to adjust to a changing oxygen level in the small intestine, ensuring it can continuously produce energy. This adaptation is a key factor in its ability to survive and proliferate in intestine environment of the host.
Moving on to the habitat of Giardia duodenalis, where it is found mainly in the first two parts of the small intestine, especially the duodenum and jejunum where the trophozoite stage is. It is the only protozoan parasite found in the lumen of the human small intestine. It also connects to the intestinal mucosa with ventral adhesive disc. This region offers the parasite readily available nutrients from food, as well as a damp area suitable for its survival and replication. The certain resident stage, known as the parasite cyst, can endure and remain dormant while outside the host in soil, water, and various surfaces for long periods of time. This greatly aids in decimating the parasite. Both trophozoites and cysts can be found in feces, which represent the diagnostic stages. The cysts are resilient and can endure in cold water for several months. Cysts can be contracted through fecal-oral route, whether it's hands or fomites, contaminated food, or contaminated water. Each cyst generates two trophozoites, which are released during a cystation in the small intestine. Next is the reproduction process of Giardia duodenalis, which gone through a sexual reproduction process via binary fission during trophozoite stage inside the host small intestine. It initially starts with mitosis, begins in both nuclei simultaneously, ensuring equal genetic material is passed on. The flagella consists of 8 per trophozoite. The adhesive disc, cytoskeleton, and other internal structures are also duplicated, as well as the ventral disc, which is used to adhere to the intestinal lining. The cell elongates and divides along its longitudinal axis, eventually resulting to 2 daughter trophozoites, followed by a cystation where it is cyst formation that starts when trophozoites move from jejunum to the colon, triggering the changes in pH, bioconcentration, and dehydration. The organism then undergoes cytoskeletal alterations, creating a hard outer layer composed of substances like chitin. It is important to note that due to physical harm to the intestinal lining, rapid asexual reproduction contributes to giardiasis symptoms such as diarrhea, bloating, and malabsorption.
Now, we move to chapter 3 where we look through what are the significance of Giardia lamblia. Giardia lamblia or Giardia duodenalis can cause diarrhea that could affect both adults and children. It has been a major public health concern in both developed and developing countries. Frequently, Giardia infections are caused by ingesting contaminated or unclean water, for instance, wells, swimming pools, lakes, and rivers. The infections also link to childcare, particularly when involve children with diapers and close contact with family members that had giardiasis infection, which this can unintentionally contaminate the spas and pools. Furthermore, Giardia duodenalis also can be transmitted through raw produced foods that is washed with unclean water, or if the food handlers who have giardiasis infection did not wash their hands properly. An individual can get infection from Giardia lamblia if their hands come in contact with dirty feces, like when changing child's diapers. Also, Anal intercourse also could be a way for Giardia duodenalis to be spread. According to a recent study, 45.3% of foodborne diseases are due to be caused by various protozoan species, with 11.2% of Giardia intestinalis are accounting in these cases. This shows that Giardia lamblia is one of the most terrible cause of diarrhea.
Thank you, Alifa. As for the last part, which is Chapter 4, example of commercialized product or process of Giardia duodenalis, I will elaborate about two examples, which is immunorun antigen detection kit and immunofluorescence assay or DFA. These two things is undergo process where it use Giardia and produce a product to commercialize. Next, I will explain more about this example. First, we have immunorun antigen detection kit. It is used to detect Giardia AG in dog or cat feces. It uses anti-Giardia antibody to detect the material of Giardia in dog or cat feces. The picture below shows the immunorun antigen detection kit that being sold. This kit is similar to pregnancy test because it functions like pregnancy test. This is because it has a band that can detect the Giardia and the band will appear purple if the result is positive. This kind of kit is being commercialized and used by pet clinic to detect the microbes in their pet feces because it gives a rapid result. Lastly, direct immunofluorescence assays or DFA. This kit is to diagnose Giardia duodenalis infection. The picture beside shows the procedure of immunofluorescence assay. It uses Giardia protein to create antibodies. It works when sample is fixed on a slide, then fluorescence antibody bind to the target antigen. Lastly, we will observe under a fluorescence microscope. Even though these two products look similar, but they have a difference between them. More specific about their function is that immunorun antigen detection kit is to detect a specific antigen, for example, Giardia in a stool, while immunofluorescence is to detect antigen or antibody using fluorescence label antibody. That's all from us. Thank you.